So now for a little bit about scallops. The bay scallop Argopecten irradians is a bivalve. That means that it has two shells. The upper shell is usually a dark mottled color, and the lower shell is white, although there are yellow and orange color morphs, and some of you will actually be receiving, we have some yellow color morphs that are part of the scallops that we'll be distributing to you. One of the first things that you'll probably notice about scallops are their blue eyes along the edge of the shell. They use these to detect movement and detect predators, and they can have upwards of 40 of these little blue eyes. Another thing you'll quickly notice about, scallops, if you go scalloping, is that, there we go, that they're quick swimmers. They do this by clapping their shells together rapidly using their adductor muscle. And this is what a scallop looks like when you open it up. Scallops are hermaphrodites. That means they have both male and female reproductive organs. They can reach a shell height of about 3.5 inches, although in Florida they're generally around 3 inches maximum. And unlike clams and other bivalves, with the scallops, we only eat the adductor muscle for the most part. Generally, people don't eat all of the soft tissue. All right, now for a little bit about bay scallop reproduction. The scallops are broadcast spawners. That means that they release egg and sperm in the water, and the larvae develop within 36 hours of fertilization. Larvae are pelagic. That means that they drift in the water column, and on average they drift for about 10 to 14 days. Spat will settle out of the water column at less than 1 millimeter in size. We call them spat. They're just really small baby scallops, and like Savannah said, they attach to seagrass blades, so that's one of the reasons seagrass is very important for scallops. They then will settle out to the bottom, and scallops typically live for about 1 to 2 years. Scallops are filter feeders. That means that they open their shells and filter out particles of decayed plant matter and phytoplankton, and that's what they feed on. In contrast, there's a lot of organisms that feed on scallops, like octopus, snails, rays, crabs, and of course us. And they have a couple of defense mechanisms. One is that they clamp their shells shut tightly to prevent predation. Another is that they are pretty well camouflaged in some environments. Here you could probably not even distinguish the scallops. In other environments, they're not as well camouflaged. And another mechanism of defense is to swim away, which I showed you earlier. All right, I think we've said it a few times, but seagrass is very important habitat for scallops. In addition, they also need good water quality because they use their gills to absorb oxygen from the water, and they also are filter feeders. So good water quality for scallops is a salinity that's between 20 to 35 parts per thousand. And you'll be using your hydrometer to measure the salinity in your area. This picture was actually taken by one of our scallop sitters of the hydrometer that we sent. The temperature range that they can tolerate is between 50 to 90 degrees Fahrenheit. And there are many threats to scallop populations. Red tide is not good for scallops. Pollution, large rainfall events, and high turbidity, which is a lot of sediment in the water column. All of these things can negatively affect scallops. Scallops used to support a very large commercial fishery from Florida Bay to Pensacola. Sorry, I don't know why it keeps jumping ahead like that. This graph shows the pounds of scallops harvested from 1930 to when the fishery was closed in 1994. And you can see that the pounds harvested declines rapidly in the late 60s, and the fishery was actually closed in 1994. Some causes of fishery collapse might be habitat loss, diminished water quality, overfishing, and recruitment failure. From 1994 until present, the bay scallop is a purely recreationally fished species. This is the season structure for this year. You can see that there are 5 regional zones from Pasco County in the south up to Gulf County in the north. Last year, the FWC Marine Fisheries Management held a series of workshops where they asked for public input, and they used this public input to set the season structure. Now, if you're interested in going scalloping this year and you want to go near you, the nearest place is Gulf County, and the season is from August 17th to September 30th. For about the last 25 years, FWC scallop biologists have been conducting annual surveys. They conduct these surveys at the locations shown on the map here, although those locations have changed somewhat over time. And they conduct the surveys by diving or snorkeling and counting the number of scallops that they observe along a transect. That's that yellow line in the image there. They lay out a 300-meter transect and count all of the scallops observed within 1 meter on either side of the transect. They also record shell height of all of the scallops that they come across. So that gives us an estimate of scallop density and size structure at each of these sites. So we have a long-term data set. These data are from this long-term data set collected by FWC scallop biologists from 1994 until last year. Scallop density per square meter is given on the y-axis. And if the scallop density is greater than 0.1, that means we have a sustainable population. If it's between 0.01 to 0.1, it's vulnerable. And if it's less than 0.01, the population is considered unsustainable. So if we overlay that on the graph, what you can see is that these populations are frequently in the vulnerable area, rarely unsustainable, and frequently they are in the sustainable range. So these two populations, steenhatchee and homosassa, are considered the core of our population in Florida. In comparison, if we plot up St. Andrew Bay and St. Joseph Bay, you can see that these two populations are rarely in the sustainable range, and they're frequently in the vulnerable range and the unsustainable range. So the goal of our restoration project is to increase depleted scallop populations in the bays like St. Andrew Bay and St. Joseph Bay, and also to reintroduce scallops to other suitable areas like Pensacola, from which scallops have disappeared. This is a 10-year project that we began in 2016. The regions that we are focused on are shown by the boxes here, although we have dropped region 2. And our original plan was to start with St. Andrew Bay and gradually expand to the other regions. However, there was a large red tide event in the summer of 2015 through early 2016 all throughout the state. It also affected St. Joseph Bay. You can see there are medium to high levels here of the red tide event. And so we had very little recruitment, and as a result, you can see here that the population of St. Joseph Bay in 2016 was at an unsustainable level. In 2017, it just barely inched up to vulnerable, but it's still not exactly where we want it. And so we had a change of plans, and we decided to focus on restoring St. Joseph and St. Andrew Bay, and then gradually expand to the other regions. Now, how do we restore scallops? Well, one of the first things we do is to collect brood stock from St. Joseph Bay. That's a picture of some of our brood stock, and we provide this brood stock to a hatchery, the Bay Shellfish Company, and later they provide us with larvae or spat, and this is a picture of some of the scallop babies that they have provided to us. We also collect wild spat. We use these traps shown here. There's a cement block attached to essentially a citrus bag. The scallops settle into those bags, and then we collect them. We then raise both the hatchery and the wild scallops to a size of about 10 millimeters, and those are the scallops that you guys will be receiving today. Last year, we placed these scallops in predator exclusion cages in St. Joseph Bay, and the expected result is that the cages will likely increase increase the number of offspring produced. Now, you may be wondering, how does holding scallops in cages actually increase offspring? And I'm going to tell you now. All right, so as I mentioned, there are relatively low scallop densities in St. Joseph Bay and St. Andrew Bay. So when these scallops spawn, there's a fairly low chance of fertilization occurring, which means that there are no scallop babies that year. However, if we put scallops in cages, first of all, we're protecting them from predation, because remember, everything loves to eat scallops. And that way they're more likely to survive to reproduce. And also, when these scallops spawn, there's a higher fertilization success rate. And so then we have more offspring, and that's how we restore scallops in the bays. So last year, as I mentioned, we placed about 3,700 scallops in cages in St. Joseph Bay and about 350 wild scallops. And this year, we reached out to the communities of St. Joseph Bay and St. Andrew Bay, and we have had an incredible response from you guys. So we have had 2 cages are going to be maintained by volunteers, about 150 in St. Andrew Bay and 50 in St. Joseph Bay. And this was just based on the response that we got, not any planning on our part. And we're planning on giving each volunteer about 25 scallops, so that would give us about 5,000 scallops that would be maintained by our volunteers. Our FWC biologists will also be maintaining their own cages of scallops, so combined, this effort will nearly double our restoration work this year. So again, a very big thank you for showing up and for volunteering to be a scallop sitter. These are the locations of the bay and dock cages in St. Andrew Bay. You'll notice that at some of the locations, there are multiple cages, and that's because we have organizations like the AMI Kids organization that have volunteered to take multiple cages for us. And these are the locations in St. Joseph Bay. We provided many of you with hydrometers, and we received some salinity data from you. So Austin plotted this up, and you can see that there's a wide range of salinities in St. Andrew Bay and more consistent salinities in St. Joseph Bay. All of this information was from you guys, our volunteers. All right, I just want to remind you that when you agreed to become a scallop sitter, you agreed that you would clean your scallops once a month and the cage that they're in, and to take a salinity reading with your hydrometer. We would like you to record some information in a shared Google document that we'll send to you after the workshop. Every time you clean your scallops, we'd like you to record your name, the date that you cleaned your scallops, whether or not you cleaned your scallops, the number of live scallops that you observed, and you can tell if they're dead if they are popped open and they stink. That's a good indication that they're dead. Record your salinity and your start and end time for how much time you volunteered. You can take multiple salinity measurements using your hydrometer. In that case, you would just say that you didn't clean your scallops and just record your salinity measurement. We're asking that you clean the scallops and the cage on the 1st of the month, preferably. If you can't do that, that's okay, but we just prefer that. And you can clean your cages and scallops multiple times every month. I wouldn't recommend doing it more than weekly, but you can do it weekly. And just every time you clean your scallops and get the count recorded in the Google Sheet for us. Please start doing this next month, beginning in May, and the project will conclude in January. And we have a Facebook group. If you haven't already joined, I encourage you to do so. You can post your progress, meet up with other volunteers, put pictures of your scallops on there so that we know that they're actually still alive. These are all the materials that we'll be providing to you. The materials that are listed in black are ones that we would like back at the end of the project. The ones that are listed in red are yours to keep. If you're using a dock cage or a bay cage, you'll notice that there's a slightly different setup, and Austin will go into this in more detail a little bit later. So as I mentioned, we'd like you to return your supplies. Those supplies should be returned to us in February of next year. However, this is going to be an ongoing project. So if you decide that you want to keep raising scallops, then in February, let us know, and you can hold on to your supplies. However, this is contingent on active participation in the program. You need to be actively posting your information in the Google Sheet, and also you need to have 5 scallops alive at the end. So I hope that you will continue being a volunteer with us. And also at the end in February, you will release your scallops into the bay. All right. And so with that, I'm going to hand it off to Austin, and he will provide you with a demonstration on how to clean your scallops and cages.
So I feel like I know all you guys from email, but it's nice to put a face.
I just forgot about this scallop rodeos
Go.
part. I'm sorry. Psych. All right, we have this other restoration project that we might do called Scallop Rodeos. And in the Scallop Rodeos, anyone who's interested in scallop restoration will collect wild scallops in either St. Andrew Bay or St. Joseph Bay. This will be on one day. We'll limit this to 50 scallops per volunteer, 100 volunteers per bay, so a total of 5,000 scallops possible. We will take the scallops and place them in cages and maintain them ourselves. And in St. Andrew Bay, we will limit the area of collection. So we're just looking for feedback on this project. If you have any questions or suggestions, please let us know. You can provide written feedback. We have some forms in your folder. If you would prefer to provide us with written feedback, you can do that. But just by raise of hands, are there people that support this idea? All right. Are there any people that do not support this idea? And are there any suggestions or questions about it? Yes.
You just think it would be important to spread out so you don't take them all from one area?
Yes. In St. Joseph Bay, the collections will be open to the entire bay. In St. Andrew Bay, for some reason, I can't pull up the map here, but it would be limited to, never mind. All right, so now I'm done. Sorry, Austin. Take it away, Austin.